All right. Good afternoon to everyone on Teams, all 80 of you, and then 11. Goodness, no, 10. 10 people. Auditorium. Fantastic. All right, so hopefully everyone survived test block, and then also at the same notion, hopefully everyone put strong effort into this practice test that we gave you all for this Friday. I did see that there were around 26 people that opened the test and didn't answer any questions. So for those of you that are doing that, I would try to take the time to answer them because this is your chance to test yourself to see if you're getting the knowledge. If you just open it and don't answer any of the questions, that will come back to bite you. All right, so for the very first micro question, a patient comes into your clinic with complaints of watery diarrhea after eating at a fast food restaurant. The diarrhea then progressed to bloody but never showed a fever. What is the most likely cause of this patient's condition? So the question is asking you, what bacteria are we talking about? If you look at our specific, and I didn't share the screen. There you go. No, it's not your fault, Ed. Just complaining and bitching and not paying attention. All right, so the first question is asking you what bacteria this is. So then you have to look at the question stem to see some of the pertinent features. So here we have watery diarrhea, which then progresses to bloody. So that in itself should give you a hint. But if you needed more of a hint, here, fast food, so just like Dr. Anderson was talking about with burgers. If it's watery, then going to bloody, and it's from fast food, our strain is going to be EHEC, so that O157H7. That's going to be our answer choice. Like I've said before, kind of go through these and see why these wouldn't be answers. There are some here that have watery, and then there are some that have bloody, so inflammatory stool. So you have to go through, kind of figure out why each answer choice is incorrect. So the next question, you all should have noticed I used the same question stem for 3 of these. A 92-year-old nursing home man presents to his doctor with a necrotic wound on his right forearm. After taking a wound sample, test results reveal small purple grape-like clusters, catalase positive, coagulase positive, antibiotic resistance to methicillin. What virulence factor is most likely to blame? So if we go back through and look at our stems here, it's telling us grape-like and purple. So that's going to be gram-positive, cocci. Then in clusters, if it's in clusters, that means it's going to be a staph. You only really talked about one strain of staph at all. Catalase positive, another staph, coagulase positive. That should give you more idea. And then antibiotic resistance to methicillin. So here we're talking about MRSA. It was stated in the lecture that MRSA does have specific virulence factors in comparison to others, especially community-acquired MRSA, which this elderly man is in a nursing home, so that would be community-acquired MRSA. And the biggest virulence factor for a necrotic wound would be PVL. And then the same question stem, which of the following is the treatment of choice for this infection? So because it is MRSA, we know it has high resistance to a lot of antibiotics. The main choice of treatment for MRSA is going to be vancomycin. That's straight from these slides. Remember, like always, go through each of these. I tried to make sure that each of these answer choices were not terrible distractors, that they weren't completely out of the ballpark, that they were actually learning opportunities. That's something I discussed with Dr. Anderson after that wonderful review on Wednesday. So that way, when you all go through these, you can look at these answer choices and use them as learning opportunities. Then the third one from the same question stem, which clinical result would appear in this patient's blood work? So this one was a little tricky. When you go back through and you look at PVL, some of the symptoms of PVL would be leukopenia. So leukopenia being a decrease in leukocytes, so a decrease in white blood cell count. And like I said, go through these other ones and see why they wouldn't be the obvious answer choices. Really try to give a good explanation, not one that just says just because. If you guys have questions on specific ones of these answer choices on why they might be wrong. Oh, really? It does? Okay. Must be really annoying during lecture. Dr. Anderson hasn't seemed to like, does he not notice it? Oh, okay, because I was saying every lecture this week, he just keeps going, and I didn't see him notice that at all. So yeah, if you have any questions specifically on why some of these aren't answer choices or why they aren't correct, just send me an email. I'm not going to go through them today because they are long, detailed explanations, and I kind of want you guys to build those explanations because block 2 test is harder than block 1. Our next question, you travel to Mexico to explore the Great Ruins. After the first day, you experience watery diarrhea, nausea, and vomiting. Through your understanding from Dr. Anderson's lecture, you surmise the cause should be ETEC. Which of the following is a result of the exotoxin produced by the organism causing your symptoms? So here, what is the toxin and what does it actually do? So we know from ETEC, we have that the cholera toxin, which is going to give you an increase in CAMP and cyclic GMP, which is going to increase the activity of the intestinal cells, which just forces electrolytes out of the intestinal cells into the lumen, and then water follows, causing severe dehydration and very, very watery diarrhea. A 5-day-old infant is hospitalized for meningitis. A Gram stain and testing of this sample is most likely to show which of the following. Everyone did really well on this one. So here, because we're talking about a 5-day-old infant, we're talking about neonates, and we only have one bacterium at this point that has neonate meningitis, and that's going to be Strep agalacti. So the specific test results for Strep agalacti, it is gram-positive cocci in chains. We know it's in chains because it's a strep. And it is catalase negative, once again, because it is a strep. And then it's group B, which is beta hemolysis. Then I would go through these and give which these actually are. A 27-year-old farmhand starts to experience diarrhea with blood and leukocytes. After proper isolation of the microbe, the physician notes a very thin curved gram-negative rod. What disease is a potential risk factor for cross-reacting antibodies? So here, you can look at the question stem, very thin curved gram-negative rod. There's only one bacterium that's like that right now, Campylobacter jejuni. Also, other pieces in the question stem. He's a farmhand, so he's working on a farm around farm animals, which are the reservoir for that bacteria. The potential cross-reacting is going to be Guillain-Barré syndrome, which is a nervous system syndrome. That one's, once again, straight from the slides. Make sure you go through and say what scarlet fever comes from, where rheumatic fever and glomerular nephritis comes from. A water treatment plant. At water treatment plants in the United States, water is inspected for coliform as an indicator of fecal contamination. A specific bacterium was found to have very low numbers but still caused infection. The symptoms of such infections result in cramping and very bloody diarrhea. What bacteria could you assume is the cause of these infections at such a low dose? So this is a tiny, tiny little fact that he actually had in his slides and he went over. He put the infection dose of each bacteria or of each of the coliforms in there, and the lowest dose would be Shigella species. Now, the reason it's Shigella soni is because this is the highest or the most prevalent in the United States. A 9-year-old male is suffering from fever and a diffuse rash throughout the body and a unique strawberry appearance on the tongue. What is something unique to the infecting strain of the bacteria? So you need to look back and the question stem, strawberry appearance on the tongue. So that's just going to give you the answer, the largest answer right there. From there, knowing which bacterium it is, so Pyogenes, you then have to know what its actual virulence factors or toxins are. So it does have an exotoxin. That exotoxin is contained from a lysogenic phage. What is the primary supportive treatment for a patient experiencing a secretory diarrhea? Everyone did really well on this one. So IV and oral hydration with electrolyte replacement. That's going to be the biggest supportive treatment. So that's how you can support without actually giving the patient any sort of chemical or treatment that could affect production of toxins or affect the bacteria at all. And go through, all of these are explained, well, all of them except for fluid restriction are explained in your slides. So make sure you go through those. Fluid restriction, I'll explain that one. That shouldn't make any sense at all, because if you restrict fluids while someone is having secretory diarrhea, they're having very watery diarrhea, so you're just going to dehydrate them more. Untreated infections with pyogenes can result in many secondary syndromes. One of these acute glomerulonephritis is caused by blank. So I don't know if you guys have gotten to it in anatomy yet, but the kidney does have a section in it, the glomeruli. That's where you're going to start filtration. In strep pyogenes, when we have acute glomerulonephritis, it is a type 3 hypersensitivity. So it's antigen and antibody complexes binding together and getting stuck in certain places, and where they're actually getting stuck is in that glomerulus, and it causes proteinuria and hematuria. It's just going to be protein in the urine and blood in the urine. Which Staph aureus virulence factor could be considered a superantigen? So he went through this one and he explained that there are two main superantigens in Staph aureus, one of them being the toxic shock syndrome toxin 1. Going to be your TSST1. So those are mine. Does anyone have any questions regarding my practice questions?
Hey, Daniel, what was the other super antigen for Staph aureus?
Sorry, can you say that one more time? We didn't have the volume on in the lecture hall.
You had said there were two super antigens for Staph aureus. What was the other one?
I'm going to let you find that one out.
Gotcha. Thank you.
Sorry, I know it's being rough and being annoying that I'm not explaining all of them, but this was at the request of Dr. Anderson, so that way you guys go through and find the answers yourselves. It is on a slide, on one of the slides where he talks about them. He has two of them listed. He has super antigen written underneath one of them, and then he has an exclamation or an asterisk next to toxic shock syndrome toxin 1. And then it also should be on the study guide, my flowchart study guide that's on Canvas. Did I go over number 2? Okay, number 2. So number 2 with the question stem, we know it's a 92-year-old man. So that means it's going to be community acquired because he's in a nursing home, and he has a necrotic wound. So this part's really important here, the necrotic wound. After taking the sample, we see that we have the small purple grape-like clusters. So that's going to be staph. Catalase positive, once again, leading you to staph. Coagulase positive is leading you to Staph aureus specifically. And then an antibiotic resistance to methicillin. So now we know that the bacteria is MRSA. So if we know it's MRSA, and we know it's community acquired, then we have to think back to which toxin or which virulence factor would cause a necrotic wound. So the only one of these that's actually going to cause necrosis is going to be PVL, because that's a leukocidin. So that's one of the invasive ones that actually breaks down the epithelial barriers. And that's how we know that it is PVL, because it is necrotic. Anyone have any other questions? This one. Make sure when you're going through these and you're practicing them and you're using them as study tools, go through, find out exactly which answer is right and explain why, and then find out why the incorrect ones are wrong and explain why in great detail. If you do that, I made sure to have all of these answer choices cover all the information in both lectures. So if you do that, you will actually cover all of the pertinent information for both lectures for this week. Any other questions? All right, I will pass it off. If anyone has any questions, like always, you can send me an email.
You guys heard about Sketchy Micro? Yeah. Some of my friends like that. Personally, I didn't really like it that much because I felt like it was a lot to go through, but if you're having trouble remembering all of this stuff, maybe that could help, because it makes like a story about each like bug. So then it's like easier to remember the story rather than just like random facts. So that's an option. Okay. Hopefully you got the email that Dr. Blank sent out today about the exam review that's going to be on Monday. If not, there's an exam review and it's going to be on Monday. There should be a Teams link. So like just check your Teams calendar or like your Google Calendar like on your phone. You can just accept the invitation. It's going to be similar to like how Immuno did the exam review. We can't go over the entire exam because then there's no like exam security. So that sucks, I know. But at least they're doing something for you guys, because when we got switched to online, it was just like, sorry. So hopefully you guys take advantage of that. Start thinking of like your questions now, because maybe if you're not just put right on the spot, maybe you can like think about certain questions that you were confused on from the exam. So yeah, okay. Our first question, what transcription factor phosphorylates RNA polymerase 2 in the pre-initiation complex? A lot of you got this right. It's going to be TF2H. TF2H has helicase activity, which we learned about last block, and now we're learning about its kinase activity. So RNA polymerase 2 enters this pre-initiation complex first, but it's not active until this TF2H phosphorylates it. True or false, most of the general transcription factors interact with the DNA. A lot of you guys got this right. That's false. The general transcription factors interact with themselves, not with the DNA. So like, for example, the TAFs are general transcription factors, and they bind to the transactivators, not the DNA. So the two things that bind to the DNA specifically are going to be the transactivators, because they're binding that specific regulatory sequence on the DNA. And then the TBP portion of TF2D is binding to the TATA box. So those are like the two things that are binding to the DNA. Everything else is like binding to the other general transcription factors and proteins that are involved in transcription. So if I were you, I would go through and make a list. What are the general transcription factors? What are the specific factors? What is their action? And are they binding to the DNA, yes or no? Because he will test you on if they're general or if they're specific, and he will test you on if they're binding to the DNA or not. So make sure you know that. All of the following are examples of general transcription factors except for. So that's going to be transactivators. All the rest of these are general transcription factors. Transactivators are going to be specific because they're binding to those specific regulatory regions on the DNA. So on slide 8, you can find your list of general transcription factors. So go there, make your own notes on it. What do they do? Are they binding to the DNA? Yes or no? And then slide 15 has your specific. So go through those two slides and make like a little chart. And then practice that chart on a whiteboard from memory so that you know you're set for the exam. A viral infection results in the release of cytokines throughout the body. These cytokines bind with the cells of the body and lead to changes in gene expression and subsequently protein production in those cells. This is an example of what? So we only really talked about two of these answer choices. We talked about developmental regulation, or we talked about inducible regulation. So these other two, we didn't talk about them. Don't pick them, because he's not trying to trick you. He just probably needs two other decoy answers. Since we only talked about two, he's going to come up with two other ones. So we only talked about two, and from there, we can tell that this virus is like inducing a change in transcription. It's causing us to transcribe those genes to make those proteins to initiate a response against this virus. So that means it's going to be inducible regulation of transcription. Okay. Which of the following do co-activators recruit? I'm so happy to see a lot of you guys got this right, because hats and HDACs are not going away. We still need to remember those. So co-activators are trying to activate transcription. If we're going to activate transcription, then we need to have loose or decondensed chromatin so that we can have access to the DNA for transcription. To get loose or decondensed chromatin, we need these hats adding acetyl groups onto the histones so that they lose their positive charge and don't interact as closely with the negatively charged DNA. So it has to be a hat. And then co-repressors recruit HDACs because we know HDACs remove those acetyl groups, regain its positive charge, interacts closely with the negatively charged DNA. Okay. So transactivators can directly bind to all of the following except, okay, this one was a doozy, I guess. So transactivators are binding to that regulatory element. That's like what they're looking for. That's where they're going to land on that DNA. We know TAFs also bind to transactivators to try to stabilize that complex. And then transactivators can recruit hats themselves, or they can have those co-activators recruiting those hats. So those two are true, but it does not directly bind to RNA polymerase II. During development, one parent cell can result in daughter cells with different gene expression levels due to what? That's going to be that asymmetric distribution of transactivators in the parent cell so that each daughter cell winds up with different amounts of different transactivators. So what's our main reason for having a transactivator? To induce transcription of certain genes to make certain proteins. So we start from like one embryonic cell, and then we're like going through cell differentiation. We're making different cells like kidney cells, like eye cells, whatever. So all of those cells don't need to have transactivators for every gene, because my kidney cell is not going to need a transactivator to make like rods and cones, because I'm not trying to see with my kidney. So each cell is getting like their specific transactivators. So it's an asymmetric distribution specific to that cell. Congenital cardiac abnormalities typically show high amounts of mutations in which genes. So that's just something that we talked about. It's on slide 41, this TBX1 and TBX5. So just memorize that. Anytime he like brings up a disease or like a therapeutic or something, make sure you know that, because this is a medical genetics course. So he wants to relate it to medicine as much as he can to show you why we're learning the things that we're learning. And, you know, he's likely to test you on that. So anytime we talk about a disease like cancer, a therapeutic, anything, make sure you know and understand that. All right. At which point will transcription automatically occur? So transcription is not going to occur without RNA polymerase II. So we can have all the other stuff, but if we don't have RNA polymerase II, it's not happening. RNA polymerase is what's like actually carrying out the transcription. So we need RNA polymerase for it to occur. It doesn't matter if our chromatin is decondensed and it's like ready to go, we still need that RNA polymerase II to go through transcription. So only when we have RNA polymerase II will we do transcription. Okay. Which of the following are regulated in development by transactivators? So he posed this question in class. He told you it could be biochemical differences, morphological differences, both or neither. So the answer is both. The reason it's both is because we need transactivators to make those specific proteins. And for example, we can have that when we're like making our arm. So it's like, how long is our arm going to be? What's the shape of our arm? How many muscles and bones, whatever. So it's going to regulate those morphological differences and also the biochemical differences. So when we talk about like globin switching from fetal hemoglobin to adult hemoglobin, we're going to have our transactivators regulating that. Okay, true or false? NF-kappa B is a trans activator. That's going to be true because, well, number one, he said it. Number two, because it's binding to that DNA and then it's inducing the transcription of a gene. So if it's leading to transcription of a gene, it's a trans activator. In the NF-kappa B signaling pathway, I-kappa B is removed from NF-kappa B via phosphorylation. So I-kappa B is an inhibitory protein for NF-kappa B. And when we want NF-kappa B to be active, we're going to phosphorylate this I-kappa B so that it can release NF-kappa B, and then NF-kappa B is free to bind to that DNA and act as a transactivator and cause transcription of that gene. So you can think of like I for inhibitory, kappa B because it's relating to NF-kappa B. Okay, any questions? Anyone on Teams have a question?
Ivy, can you go back over 16, please? Which two did you say we didn't cover in class?
So we only covered inducible regulation of transcription and developmental regulation of transcription. Those are the only two that we talked about in class. What slide is that? Let me find it. So that's going to be on slide 14. The title is Regulation of Eukaryotic Transcription, and it says 1, developmental, or 2, inducible.
Thank you.
You're welcome. Does anyone else have a question? Wow, all right. Guess you guys really understand genetics then. All right, that's it for me. See you guys on Monday.
Hi guys. Okay. So it looks like you guys did really well on my questions, which is great. Or maybe I made them too easy. Okay. So the first question, lactase is the enzyme responsible for assisting in the digestion of the disaccharide lactose by cleaving it into 2 monomers. A person with lactose intolerance is unable to produce this enzyme and therefore unable to cleave between which 2 sugars. So pretty much you just have to memorize what lactose is made of, glucose and galactose. It's really easy because the word lactose is in the word galactose. So that's how you can remember it. Next question. Glycolysis is a sequence of 10 reactions that yields a total of blank ATP in which major product under aerobic conditions. So it's going to be a total of 4 ATP. So if I had said net of what ATP is like net, then it would be 2 ATP. But it makes a total of 4, but because it uses 2 at the beginning, it only nets 2. And that's because you take the fructose and you cut it into 2 and it makes those 3 glucose 3-phosphate that goes down the rest of the pathway twice. So you get 4 total. And then pyruvate because I said aerobic conditions. Okay, so the next question, which of the following is the rate limiting enzyme for glycolysis in all other cells and in pancreatic cells respectively? So for all cells, it's phosphofructokinase, and for pancreatic beta cells, it's going to be glucokinase. So make sure you know the difference between the 2. Following the consumption of a large slice of birthday cake, all the following would occur except, so this is just making sure you know the pathway to release insulin. So initially, your GLUT2 transporter would transport glucose into beta pancreatic cells, and then glycolysis would proceed, and that would increase the amount of ATP in the cell so that the ATP-gated potassium channel would be blocked, and that changes the depolarization or depolarizes the membrane. And so your calcium would enter through that voltage-gated channel. So voltage-gated potassium channel doesn't exist, so that's why that's false. The next question, which of the following does not contribute to the formation of acetyl-CoA from pyruvate? So your PDH complex is obviously going to be the enzymes group that, create acetyl-CoA from pyruvate. And that's made up of your dihydrolipoyl transacetylase. And that specific enzyme has the lipoic acid prosthetic group on it. So all of these are responsible for making acetyl-CoA. PDH kinase, on the other hand, is going to inhibit that and PDH complex. So that obviously would not contribute to it. Okay, which of the following will lead to an increase in isocitrate dehydrogenase activity? So this is just from memorizing that one slide with all the activators and inhibitors for the TCA cycle. So an increase in ADP would allow for an increase in isocitrate dehydrogenase, because remember your ADP is building up, so you want to make more ATP, so you want to like trigger the TCA cycle on. All of these other answers are actually what would inhibit. Which of the following are the correct products per 1 acetyl-CoA of the TCA cycle? So this is one cycle of the TCA cycle, and this is just from that slide. You make 1 FADH2, 3 NADHs, and 1 GTP. So that's just like memorization. Okay, so Dr. Patel wants you guys to understand how, like, the why behind each of these like diagnoses. So you kind of have to understand how these impact the pathway, because he's going to ask you something like more detailed to that. So you can't just like memorize it. So a 3-week-old male patient presents to the ER with severe neurological impairment. Following a urinalysis, it is determined that the patient has abnormal amounts of fumarate and elevated levels of succinate in his urine. Which of the following is likely the cause of his symptoms? So if you have elevated levels of fumarate and succinate, that means that you're going to have a deficiency of the fumarase enzyme, which is what converts fumarate into malate. So that's why it's that one. And then all the others, I think pyruvate dehydrogenase is, don't remember, but pyruvate kinase deficiency is going to be those spiny red blood cells. Okay. Oh no, the pyruvate dehydrogenase is going to do the increased lactic acid. Okay, so with an iron deficiency, which of the following complexes would be affected? So this is going to have to like test your knowledge on which complexes have use iron as like a part of it. So that's going to be complex 1, 2, and 3. And remember that complex 4 uses copper ions instead. The addition of an uncoupler results in which of the following? So it's not going to stabilize the protein gradients, because this is like equivalent to like poking a hole in the membrane, and so it just destroys the proton gradient. It's not going to inhibit complex 1, that's like irrelevant. It's definitely not going to increase ATP production, and that's because if you poke a hole in the membrane, you ruin the proton gradient and you're going to actually make less ATP. So we saw from his 3 graphs that you're going to have an increase in oxygen consumption, because that's going to cause the electron transport chain to work harder to make sure that the proton gradient is at the level it's supposed to be. And remember that with the electron transport chain, you're going to be converting oxygen into water, so your oxygen concentration is going to decrease and your oxygen consumption is going to increase. Okay, which of the following is not one of the 4 tenets of the chemiosmotic theory? So this is another memorization one. So most of you got this right. The inner mitochondrial membrane is permeable to protons. It should say impermeable to protons, and then that would be a true statement. So that's why that's wrong. And then which of the following is true regarding the electron transport chain? ATP synthase is composed of F0 and an F1 subunit. So that's why that one's false. The electron transport chain is independent of oxidative phosphorylation. No, that's false because they're coupled. Complex 1 can be inhibited by oligomycin. No, oligomycin inhibits ATP synthase. And then NADH yields more ATP than FADH, and that's true because your P to O ratio for NADH is 3, whereas it's only 2 for FADH2. And that's it. Any questions?
Phoebe, can you? Oh, you can go first.
Okay, can you go back over the question 34 for me again? I'm sorry.
Which one's 34?
That's the uncoupler.
Oh, okay. Okay, so remember that an uncoupler is going to transport like protons across the membrane. So it's like essentially making a leak in the membrane. So because your ATP synthase is going to run on that proton gradient. If you poke a hole in the membrane, it's not going to allow for this proton gradient to be like built up. So because of that, it tricks your electron transport chain into thinking that the ATP synthase is running and it needs to start building up that proton gradient. So when you turn on your your electron transport chain, it's going to yield water at the end. So that's going to take an oxygen molecule and put it together with the protons and the electrons, and then that's why you're consuming oxygen when you run the electron transport chain. Yeah, so that's why. Does that answer your question?
So you're saying the uncoupler basically makes the electron transport chain work harder?
Yeah, so it uncouples it from the ATP synthase. So like they no longer depend on each other because now you have this like other factor that's ruining that codependence.
Oh, okay. Thank you.
You're welcome.
And for number 30, real quick, did you say increase in ATP as in triphosphate is the answer?
Wait, which one?
I think it's number 30. It was the question that asked about the increase in isocitrate dehydrogenase activity.
Oh, yeah. So wait, what did you?
Oh, okay. The answer is an increase in ADP, diphosphate.
Sorry.
Okay, I just think I missed. Thank you.
You're welcome.
Hey, Phoebe,
Yes.
could you review the question about the iron deficiency and which complexes would be affected?
Oh, yeah. So the complexes affected by that are 1, 2, and 3. Because complex 4 has copper ions instead of iron.
Ah, okay, it's copper. Thank you.
You're welcome. And Layla, number 31, the one about products of TCA. It's 1 GTP. Layla, okay. And then Fernando, which one's 28? Does anyone know? Oh, okay. Thank you. There's no such thing as a voltage-gated potassium channel. There's only a voltage-gated calcium channel. We have an ATP-gated potassium channel that would be blocked. And then your voltage-gated calcium channel would be opened and you would admit calcium into the cell for that one. Any other questions?
With the coupling question again, going back to that.
Mm-hmm.
So since you have an uncoupler, essentially the electron transport chain, meaning the complexes are going to be working harder and consuming more oxygen. Does that necessarily mean that the ATP synthase is working harder, or is it just not working because it doesn't have that gradient?
Right, yeah, it would destroy the gradient, so it wouldn't be working as well because you have like a leak, so it won't be like running. Like it'll still make ATP, but like just not as efficiently.
So the complexes are going to work harder, but the ATP synthase isn't going to work harder.
Pretty much, yeah.
Okay, cool. Thank you.
You're welcome. No other questions? Okay. Justin, I think it's you.
You talk through this, right? You guys talk through this and then, okay. All right, so I have the honor of doing the last questions for you all. All right, let's see. So the first one is going to be, if you move the external and internal intercostal muscles, which muscle would you see first on the most anteromedial side of the body, closest to the sternum? Okay, so a lot of you chose innermost intercostal muscles, but do the innermost intercostal muscles go around the entire thorax region? No, right? Okay, so yeah. So since it doesn't go across the entire thorax region, I'm talking about the portion that's just medial to the sternum, it's gonna have to be the transversus thoracis muscle. Does everybody understand why that's true? Do you want me to explain, or is that suffice? Okay, so yeah, it'd be transversus thoracis, just because the innermost doesn't wrap all the way around the thorax. So transversus thoracis would be correct. Subcostal muscles are actually on the posterior side, so those are like all the way in the back. You don't have to worry about those because we didn't talk about them too much. All right, so which of the following is the most anterior impression on the right lung? So yes, that is correct. Brachiocephalic vein. Which impression would be just posterior to that on the right lung?
The trachea,
Yeah, and posterior to that?
the esophagus,
Yes, okay, yeah. And then what's the last impression?
subclavian artery.
Well, that's on the left lung. Yeah. No, archivasicus. Yeah, archivasicus. Last one. So yeah, beat. If you guys want to use that to remember it, beat to remember the right lung. Left lung is just going to be subclavian artery, aorta, cardiac notch, and don't forget that lingula too. It's important to remember. Okay. So which structure is located superior to the oblique fissure and inferior to the horizontal fissure on the right lung? That's just a fancy way of me saying like what's in between those two fissures. And yep, that's going to be correct, the middle lobe. We have the cardiac notch, which would be on the left lung. Brachiocephalic, we talked about already, and the cupula of the lung is just like the top portion of the lung. That can actually go over the first rib. All right. So next one, we have which of the following statements correctly matches the vagus nerve and the left recurrent laryngeal nerve, respectively? So yes, it's going to be D. Yeah, the vagus nerve is going to innervate most of the gut, like a majority, almost like a lot of the gut is going to be from the vagus nerve, and it's going to be in the superior mediastinum for the left recurrent laryngeal nerve. That is correct. We did pretty well on that question, so really good job. Okay, so this one's a little tricky. So the circumflex branch of the left coronary artery is compromised. Which other artery would also be affected in a typical coronary branching artery? Okay, so what's going to be, does anybody know the branches off the left coronary artery, the initial? Yeah, yeah, so yeah, it's going to be the LAD and the circumflex, and it kind of does like a bifurcation. So, and then what after that, what does the left circumflex give?
Left marginal.
Left marginal, right? So if you were to sever the left circumflex, what would happen? Which ones would be compromised? Left marginal, right? And LAD would not be compromised, right? Because it bifurcates at the left coronary artery. Does that make sense? Okay. I'm assuming since you guys are saying yes, I'm assuming they're saying yes. I'm gonna be like, all right, we're good. Okay. So if there was a blood clot that traveled into the heart and passed through the tricuspid valve, which structure would the clot pass through next? Okay, this is kind of like following blood flow. So if you go past the tricuspid valve, where are you right now?
Right ventricle.
Yep, in the right ventricle. So then the next structure that you would pass would actually be the infundibulum, which is the conus. That's that little smooth structure right before the pulmonary trunk, right? So infundibulum would actually be the first structure you see, and then the pulmonary trunk. Which one's after that, the pulmonary arteries or the pulmonary veins?
Arteries.
Yes, pulmonary arteries, right? And then obviously pulmonary veins will eventually come back from the lungs. So yes, that's why the next answer is going to be infundibulum, also known as the conus, right? All right. I try to make these questions a little bit trickier because I know you guys are telling me about Dr. Musley's questions, so I try to make them as kind of like you know, similar, I guess. But for those questions, that's why this one was going to be infundibulum. So for the next structure, which of the following pass through this structure? So first you have to identify this. What structure is this?
Coronary sinus.
Coronary sinus, right? So if you know it's the coronary sinus, then you know it's going to be venous blood from the heart. Which other two things drain into the right atrium? Anybody here? Anybody online say anything?
IVC, SVC.
Yeah, right. So SVC and the IVC, right? So, and they're going to send deoxygenated blood into the right atrium as well as the coronary sinus. So that's there right there. So you can see the superior vena cava. If you can see my mouse is actually right there, and the inferior vena cava would be right here. You can't see the holes because the angle is not obviously too good, but this is going to be the coronary sinus. Really good job on that one. All right. So, so during systole, which structures are opened and closed respectively? So is systole contracting or relaxing?
Contracting.
Contracting, right? So if you're contracting, that means you're sending blood away from the heart, right? You want to send blood away from the heart, yeah, on both sides. So if you're sending blood away from the heart, you want to make sure the AV valves are closed, right? So the AV, which is going to be the mitral and the tricuspid valve, because if those are closed and the aortic or the pulmonary valve is open, you can shoot blood out of the heart, whether it's out of the aorta or it's out of the pulmonary trunk. So pulmonary trunk and aorta have to be open. AV valves have to be closed, right? So anything that would align like that, which would be D, right? Aortic valve would have to be open, and then the mitral valve is going to be closed. All the other ones are incorrect. Nice. All right. So which of the following is not a part of the left upper quadrant? So yes, a lot of you got that. Really good job. Sigmoid colon. What quadrant is that going to be part of?
Lower left.
Yeah, okay, yeah, so lower left, yeah. This would be lower left quadrant. Stomach and spleen and jejunum are mostly going to be in the the upper left quadrant. So yeah, I highlighted the highest yields of those on the notes this morning. So if you didn't go, we'll talk about it at 5 p.m. as well. The highest of yields in each quadrant. All right. So which of the following structures becomes the internal spermatic fascia? I kind of asked this question on purpose because it's not internal. It goes external oblique and then external spermatic fascia, but it doesn't go internal oblique, internal spermatic fascia. So I didn't want you to get confused thinking, oh, internal. It's actually going to be internal spermatic fascia from the transversalis fascia. What does the internal oblique actually turn into though when you go into the scrotum? We have external oblique to external spermatic fascia, internal oblique into. I know this is new information, but I'm just trying to get your mind thinking. It's super new. Say it, don't be afraid, like it doesn't, like. Yeah, so yeah, the cremaster muscle, yeah, it's really good, yeah, so good job. So it's external oblique giving the external spermatic fascia, internal oblique is going to be giving the cremaster muscle, and then you have the internal, the transversalis fascia giving the internal spermatic fascia. So yeah, it's a very good job. And remember, the rectus abdominis as well as the transverse abdominis do not contribute to the spermatic cord or like any types of descent into the testes. They only stay in the abdomen, right? So that's important to know. Really good job on that one. Okay, so if you were looking at the posterior side of the abdominal wall, which structure would be on the most lateral side? Okay, so posterior side of abdominal wall. So it's basically like if you were like looking inside of the abdomen, like kind of like inside of the body, obviously, if you were looking inside, posterior side of the anterior wall, then you're looking at the folds, right? That's what I was getting at with this question. I know it's a little tricky, but anterior wall, posterior side, meaning you're looking at the folds. It's like the median, the medial, and then the lateral folds. I didn't give you the options, obviously, because then I would have given it away, but the posterior side on the abdominal wall is going to be those folds. So the most lateral thing on the posterior side on the abdominal wall is going to be the lateral fold, which is going to be the inferior epigastric vessels, right? So I get why some of y'all said external oblique, because that's like on the lateral side, but it's going to be the posterior side of the anterior abdominal wall, right? All right. And then which of the following structures commonly passes over the ureter? This is going to be the water under the bridge. And then, yep, it's going to be correct. The two answers above going to be uterine artery as well as the vas deferens, right? All right. Any other questions? I think that's it for me. Yeah.
Justin,
Yeah.
could you review the question again about the most anterior impression on the right lung? I missed the answer for that one, sorry.
Yeah, I got you. Brachiocephalic vein.
Perfect, thank you.
Yep. Can you show the answer for number 39? Which one's that? I can't see numbers. Middle lobe, okay. What, can you show the answer? Yeah, it's middle lobe is going to be the answer for that one. Okay, so for 47, would the external oblique be covered by deeper muscles? 47. Oh, second last one. Thanks. So with the external oblique, what are you asking? Like, are you talking about like in the abdominal cavity view, or are you talking about in the spermatic cord? Well, regardless, it's going to be the most superficial one.
Like, since we're looking from the posterior side at the anterior wall, would the external oblique, you just wouldn't be able to see it?
Yeah, you won't, no, because the peritoneum is the first thing you're going to see.
Okay, thanks.
Gotcha. That makes sense, right?
Wait, so can you explain the view that we're supposed to be picturing in our head for that question again?
Yeah. So you have like your anterior abdominal wall, and it's kind of like, okay, if you cut somebody's body in half and you look at the inside of it and you're looking at the anterior abdominal wall, that's the view you have to get. Does that make sense? Like you just cut the body in half and you're looking inside of it, that's the posterior side of the anterior abdominal wall. Does that make sense?
Yes.
Yes? Okay, so like I cut my body like straight down like this, and then like you just throw away the posterior side of me, and you look inside of the body. When you're looking inside of the body, the first thing you're gonna see is the posterior side of the abdominal wall. Does that make sense? Am I explaining it well or no? Yeah, okay. Okay. Does that make sense? I think Destiny, did you ask me that?
Yeah, so, okay, yeah.
Are you sure?
I'm confused. So you're saying there's a posterior wall to the abdominal cavity as well?
Well, there is a posterior abdominal wall, but I'm talking about, which is like your quadratus lumborum, your psoas major, that's like your posterior abdominal wall. But the posterior side of the anterior abdominal wall is the back side. It's kind of like the back side behind your like abs and stuff. Does that make sense? Behind your peritoneum?
Yeah, that makes more sense.
Okay, gotcha. Another little wordy question. Okay, so can you explain 44 about systole? Is that the systole one? Yeah. So basically during systole, you're gonna have contraction, right? So diastole is gonna be your relaxation. So when it's like blood pressure, it's like your 120/80. The 120 is your systole, and then the 80 is your diastole. Like, so contraction is gonna be anything that you wanna like push away from the heart. So for example, what happens like when blood goes into the, let's just say the left ventricle, when you have blood in the left ventricle, if you wanna go through systole, the mitral valve is gonna be closed, right? And then the aortic valve is going to be open in order to push all that blood out into the aorta, so you can send it out to the rest of the body. It's the same thing with the right ventricle. If you're going through diastole, it's just blood slowly flowing from the atrium, just going into the ventricle naturally. So for example, the left atrium bringing blood and dropping it down into the left ventricle, that doesn't require too much work. That's kind of like the ventricle's relaxing, which is why it's called diastole. Does that make sense? Does that make sense? Okay. Sometimes I'm trying to think about articulating it correctly. Okay. Your question? This heart picture? Oh yeah. So she asked if I can explain the heart picture. So this is going to be the right atrium right here. So this is the right atrium. Right here is going to be the superior vena cava, and this is going to be the inferior vena cava. And this is the coronary sinus. All this venous blood is dropping into the right atrium and eventually going into like the right ventricles. But these are going to be the three holes that are going to bring venous blood into the heart, right? The coronary sinus actually brings venous blood from the heart back into the heart. The inferior vena cava and the superior vena cava bring venous blood from the body to the heart, right? Does that make sense? Okay, perfect. Anybody else? It can be about anything. It doesn't even have to be about this.
I have a quick question. Since this is the last monthly lectures, will the next quiz be a little bit easier questions in general then?
Yeah, Dr. Werenowska is pretty straightforward. So, I mean, I'll try to get those questions aligned to like what I feel like would be relevant to the exam. So I'll make those a little bit more straightforward. We didn't have Dr. Lopez last year, so I might just go towards the harder side to be safe than sorry. So I'll go towards that. But Dr. Werenowska is pretty straightforward with her exam. Doesn't mean not to study, but you still have to study just as hard. But yeah, I'm going to try, because I know this exam was, looking at the statistics, it was really rough. But a lot of you asked me to make it harder, so I tried to make it like harder. So hopefully it helped too. I know the statistics were a little rough, but that's okay. This is just like the quiz. It's just to see where you're at. You know what I mean? Obviously, the third and fourth lecture is so brand new. So don't worry, don't stress too hard. Obviously, just catch up on the material this weekend, and then you all got this. I believe in you. Can you repeat the two choices for the last question? Yeah. Okay, so for the two choices, so you have the water under the bridge. So if you didn't go to my session in the morning, but water under the bridge is basically, you have to picture the ureter as being water. Obviously it's urine, but you want it to just picture it as being water. Just say like you're super hydrated. It's basically water. So water under the bridge, meaning water is going to be the ureter, and the bridge is either going to be the uterine artery in females or the vas deferens in males. Obviously they don't do the same thing because one's an artery and one is obviously for like sperm. It's completely different, but they are going to be over the ureter, right? So that's what water under the bridge means. Does that make sense for the two choices? Yeah. Okay. Anybody else here online? Any questions?
Justin, for the infundibulum question, are you basically traveling in the path of blood to the lungs? Like, how does that come into play in that pathway, or am I mixing things up?
No, you got it right. It's the blood is only going to flow one way. So when it goes from the right atrium, so when I say passing the tricuspid valve, it's going from the right atrium to the right ventricle, and then the next thing it's going to go is into obviously the pulmonary trunk, but the structure before that would be the infundibulum, pulmonary trunk, and then pulmonary arteries, and then et cetera to the lungs and back. Does that make a little bit more sense? Do you want me to clarify more?
Can you please clarify more?
Yeah, so basically, so you have your superior and inferior vena cava as well as your coronary sinus all dumping into your right atrium, right? That's all the venous blood going to your right atrium. Once that happens, that's going to go past your tricuspid valve and go into your right ventricle. Right ventricle is going to pass your infundibulum, which is like the smooth area, into the pulmonary trunk. Pulmonary trunk is then going to go into the pulmonary arteries and then go towards the lungs, right? This is still venous blood. It's still not oxygenated because it happens in the lungs. Once it gets into the lungs and you do, obviously, capillary exchange, alveoli, et cetera, it's going to come back through the pulmonary veins into the left atrium, and then it's going to go through the mitral valve into the left ventricle, and then out the aortic valve. And then that's oxygenated blood. So the left side of your heart is always going to have oxygenated blood. The right side of your heart is always going to have deoxygenated blood. You don't want any switching in between.
That makes sense. Thank you.
Yep, gotcha. What exactly is the significance of the infundibulum? I wrote down this thin area between. Honestly, there's not that. It's probably just like an embryological like remnant. It doesn't have too much significance. They just noted it because it's a smooth area on the right ventricle, so it's just kind of odd. So they just gave it a name because of its area. It's kind of the same thing as like the Fosso Valley. I mean, it did something before, but now it's just like part of the wall. So it's kind of like the same thing. They just gave it a different name because like embryological development probably. Yeah, gotcha. Anybody else? Is that a question or no? All right, sounds good. All right, if nobody has any questions, then I'll end the team's meeting and stuff like that, and then just obviously just let us know if you need anything, and yeah, good luck.
Anyone on Teams have any general questions? You guys have not a lot of questions today. Yeah.
Dr. Muffler, he's only going to ask us 2 questions. I didn't go back and look at the whole material last year. I'll perfectly because that like if you spend so much time on the first block material, you're definitely going to be like second block, which is way more important. What I say is just like hammer on the second block. If you get those 2 questions wrong, they were bonus questions anyway. Hopefully you can bank on your prior knowledge to get them, but don't bank on studying the first block with 2 questions. Have 50 questions that are going to be. Yeah, so don't. And if I just throw it in there sometimes, like about like asking like serratus anterior, just to like don't go back and like. Yeah.
If anything, I would just like the morning of like a big topic like that, just like skim it. But like he's saying, don't put all your energy into that. Whereas genetics, I kind of dropped a bomb on you guys that the last test is like a final. It's not like a cumulative final, but all those topics just build and build and build. So like you see how we're talking about hats and stuff like that, talking about transactivators again. So he's going to pull questions like from each exam block and put it on your like last exam. So like I remember last year we found out that it was like a cumulative final and everyone was freaking the F out, but it wasn't like that big of a deal. It was like stuff that's like, oh, I needed to know this anyway because the information is building. So like those topics, like if you didn't get them down for the first exam, make sure you're getting them down now. Not gonna go away. Have a great weekend, guys. Oh yeah, because you have.
Wow, that one's a multiplier.
This question.
Still 14 of them?
Yeah,
Did they ask me questions? Ooh,
Oh,
are these questions?
no, yeah, that's from.
Okay. Do. Almost does the same thing, and she has it all like color coded and everything.
Oh, really? I just highlight when I'm done.
That's what she does. Yeah, she highlights when she's done with it, but she has like a color marker for me and then a color marker for her
Oh,
things, and then
yeah.
like
I used
family
to do
thinks it's
that.
crazy. She has so much stuff.
I used to do that until 2020 ruined my planner of all the pens.
She had to get a new one. Yeah.
So now I'm just sticking with pencil.
She specifically bought a new planner because so much was ruined in the 2021.
Yeah.
She's like, I'm not only. I was like, but it's a nice one. She's like, it's
Too
ruined.
much
I'm like, all right.
white out. It makes it like look even more unorganized. And I'm just like.
Someone's.
so she just snapped it with her whole body weight. Me and him were just looking at each other like hospital. We're like getting dressed because we have
So
like
should I keep going with like this kind of style, or should I just remake all?
Well, I go through like, and I take
Why is this so